Hello AP Bio students, and welcome to our video on protein synthesis. This is the transcription edition. So what is transcription? Well, transcription is basically taking the code that's in our DNA and making a copy of it. Now it's not an exact copy. There's a few things that change, but it's kind of like taking a letter and transcribing it, right? Copying it. Except for in our new letter, we don't have any T's in that letter. We change all of these T's to U's. And this copy that we're going to make, if this is the DNA, this copy that we're going to make is called mRNA. And that stands for messenger RNA. So let's take a look at the larger picture here. So we are actually hanging out right here. Whoa, this is the nucleus of the cell. So the DNA never leaves the nucleus. Like in our Lego story, the blueprints never ever leave the main office of the CEO. So we have to make copies of that blueprint in order to make the machinery that we need to make proteins and also make the actual proteins themselves. So what happens is in DNA, there's sections that are used to code for specific proteins, and this section is often called a transcription unit. And that transcription unit is code for a specific gene. Code for a gene, or more specifically, it's code for a protein. So this is going to give us this is going to code for the necessary amino acids to make the protein that we need, whether that protein is ATP synthase, an enzyme that helps us do cellular respiration, or whether that protein is insulin, which enables us to control our blood sugar levels. So as you know, there's hundreds of thousands, if not millions, of different types of proteins in our body, and DNA holds a code to make all of those functions. So we've been learning a lot about all the different proteins, so know that they can't be made without this special process. So where does it all start? Well, it starts when DNA shakes its tata, all right? At the beginning of a transcription unit, there is this special code, and that special code signals to all the machinery. In this case, our machinery is going to be RNA polymerase, where to start the gene, where to start transcribing. And it's known as what's called the TATA box. So it starts with thymine, adenine, thymine, adenine, and then it's got several A's, adenines. It keeps going. This is my keep going. Okay, and so this is going to be our what we call our sense strand. This is the strand that we're copying, and our nonsense strand or missense strand is going to be the mirror image of this. So this is in, oops, sorry. So that would be T T T T dot 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 dot. So this little sequence right here is called the TATA box. Cleverly named, right? And this TATA box says, hey, RNA polymerase, if you want to copy the gene that follows me, this is where you start. And this is where the transcription, this is called the promoter region, but this is where the transcription factors begin to assemble to start splitting apart DNA and create an mRNA. So let's take another look here. I've got an image. So we have, if this is our strand of DNA, remember DNA has a direction, 5' to 3', 3' to 5'. We're going to have our TATA boxes right here, TA, TA. And then there's usually sometimes we call it a polyadenine tail, which just means a tail of a lot of adenines. So this whole region is called the promoter region right here. And this is once again saying, hey, transcription factors bind to me. So there's a lot of these different proteins which are coded for in your DNA, that bind themselves to this ta-ta box and this promoter region. So we get a bunch of these proteins, and they almost create like a brace, or they create a little seat, if you want to say, for RNA polymerase. So RNA polymerase bonds itself to these different other little proteins right here that kind of act as a little brace to hold on to it. And now we have this whole RNA transcription initiation complex. Okay, so once this initiation complex is created, then we can actually start transcription. And this right here, this arrow shows our mRNA that's going to be created. And mRNA as well is going to be created in a 5' to 3' fashion, just like DNA is created that way in DNA replication. So once again, we have our ta-ta box, which signals transcription factors to bind to this promoter region right here. So here's our promoter region. And a bunch of other proteins bond to this promoter region, and they kind of create a brace for RNA polymerase to attach. And actually, it doesn't say it here, but before RNA polymerase gets started, we need to add some ATP in order for transcription to initiate. So if we look at DNA, I'm going to make up a few just genes here. So if we look at our DNA, so if this is our DNA in the nucleus, I'm going to kind of go like this, and then this is the genes separating right here. Go like this. There we go. So if this is our kind of looks like a big mouth. And we're going to get our top one done. And this, of course, is a simplified version. This might be our ta-ta box over here. And then we're going to have our polymerase, or our transcription complex right here. So this is RNA polymerase. And in transcription, yes, we are making a copy of one side of the strand of DNA. However, that copy is a little bit different because in RNA, we don't have any thymine. So when we say there's no T in RNA, there's no thymine, and that's that nitrogenous base. Instead of thymine, we have a U, and that stands for uracil. So when we're looking at how these things pair up, if our DNA sequence, let's say it reads A, C, C, G, T, C, A, T, G, then our RNA sequence, let's make our RNA blue, is going to read not T, but U, because A normally pairs with T, but since there's no T in RNA, it's going to be a U for uracil. G, G, C. Well, we have thymine here, but thymine binds with adenine, so we can use our adenine here. G. We've got A's. They're going to, adenine's going to bind with uracil. Thymine's going to bind with adenine, and guanine's going to bind with cytosine. And this is going to continue. I'm kind of drawing over my own writing here. And this is going to continue. It's going to keep going until it hits what's called a termination sequence. And typically, you'll see on your amino acid wheel when you do practice coding mRNA code to amino acids, that the sequence AUG typically means stop. Also AUU would mean stop. So we have our promoter region, we copy a this is called the sense strand, the strand that we're copying. We're copying the sense strand and we're transcribing it into RNA or to mRNA, and we keep going until we hit a termination sequence. And when it's done, this mRNA strand kind of just takes off and leaves, and it's going to leave the nucleus. It's going to go through the cytoplasm. So mRNA to cytoplasm, and it's going to find a ribosome, and that's where translation is going to happen. So in a nutshell, let's do a quick review. So transcription is the process of copying, and it's not exactly copying, right? It's taking a strand of DNA and it's basically transcribing it into RNA language, which has a U instead of a T. And this all happens in the nucleus. And this copy that we make is called a messenger RNA, an mRNA. So this is our photocopy of the blueprints that we need to make those Legos, right? And in order for this to start, we need a ta-ta box. DNA shakes its ta-ta, and that initiates the transcription by signaling the binding of RNA polymerase to the promoter region. And the mRNA leaves the nucleus and travels to a ribosome. So in a nutshell, that is transcription. There is an animation video that's a little more complex and we need to look at, but however, it gives you a good visual of what's going on. So I suggest you watch that after this video. Hope you're having a good day. Take care. Bye.